Okay, we walked through the three big processes that are involved in taking heterogeneous RNA and maturing it to what we call full mRNA. We talked about capping, talked about poly A tail, and we talked about spliceosomes and the splicing process. Now I want to talk a little bit about what can happen both in a positive way and sometimes in a negative way relative to splicing. In the positive way, remember that we've got only 20,000 genes or so in humans, these protein production genes. Yeah, we've got over 100,000 proteins and maybe more that clearly are functional. And you've seen lots and lots of proteins that are involved even in the process of going through transcription and the process of maturing. So you can imagine how many protein parts have to be made. Well, one of the ways we can get multiple proteins out of a single message is through alternate splicing. And alternate splicing, as I said, can be a good thing, producing these products that we need. And it also can have a detrimental effect if the splicing goes wrong. So we think about this central dogma, if you will, of molecular biology is that you have a gene. The gene produced a message, and that message produced a protein, right, through the process of transcription and then translation. We knew early on that this wasn't right. That's why protein is in quotes here. We don't make a protein from a message. We make a polypeptide from a message, and that polypeptide may or may not immediately form a functional protein, or it may have to be highly modified by its own splicing and cutting and modifications to make a protein, or it may take two or more of those polypeptides together to actually finally make that protein, as it has in hemoglobin that we've looked at before, which requires 4 units of 2 of each of these polypeptides from different genes to function. So this is, we already know this is not a direct lineage here, but the basic idea is that somehow we do go from genotype, a gene, to phenotype, which are proteins. Okay. Now, what alternate splicing means is that we can take this one message produced from one gene, right? And we end up making multiple proteins. The way that that happens is that the splicing involves different ones of these exons. For example, just in protein A here, we need all the curves and points. Maybe that's a transmembrane area or something. We need all that. So we might use exon 1, 2, 3, 4, and 5. Okay, they're all spliced into one. It could be that we skip over one, maybe this big exon 3 for some other area. Maybe that's what's needed for across the mitochondria or someplace else instead of across the nuclear membrane. And then maybe for the cellular, the outside of the cell or some other place, you need something else. So you include 3, but you don't include 4. So all these ideas can be produced, and we can do alternate splicing. This is a common way known as exon exclusion, exon skipping, where we skip over an exon. In this case, we skipped over exon 3 and used 4 and 5. Here we use 3, but we skipped over 4, so it's just exon skipping. Now what this does, of course, is increases genetic diversity. By allowing this to happen, we can end up with a lot of different types of protein products that are very similar, but have different characteristics because of the bends and functions, because of their size, because they include or disclude certain types of these functions. Okay. Now some examples here. Here we've got another one: chromosomal DNA, primary transcript. We might make one that involves all. We might skip some, etc. By far, the most record holder that we know of is in Drosophila. The discum protein variant, the one message can end up making 38,016 or so currently, I think, or so splice variants. That's more splice variants. It's more different types of final message than the entire, that's twice the number of genes that are actually found in the entire Drosophila genome. So obviously it's a very, very important outcome that we can have all that splicing. And what we've learned recently is that a lot of this is controlled actually during the process of transcription. So this process of transcribing and then this co-transcriptional splicing, a lot of what's going on here about choosing which of these exons to use or not use is really being driven by the transcriptional process itself and the activities of that. Remember back when we defined a gene and I said, okay, here's the transcriptional unit, and then we have all these we have CATA and CAT boxes, and then I showed a whole bunch of different regulatory regions that were up front of that. And I said, don't worry about them too much, but there are these different SP1s and all these others. Well, depending on which one of those units gets activated, can control and signal which one of the exons actually get used in the splicing. So there's a direct relationship between those large control units that are on genes and what actually finally comes out as the final mature message here. And we're learning more and more about how the interaction occurs between those regulatory elements, those motifs that are there, what sets down and either locks down or activates those motifs, and how they correspond to whether or not exon 3 gets used or not in a particular case. So it's a very interesting and very active area of discovery, and people are really investing some time. And now that we can do entire genomes and we can find these gene regions very effectively, and the control regions for them, and trying to understand, okay, we've got all these messages, can we backtrack now and figure out how we ended up with that message based on the fact that this gene was being read and transcribed. A very important area that are currently very active in molecular genetics. All right, just a couple of examples for us. Here's an example that is a real world example. We have a gene, we do transcription, we end up with 1, 2, 3, 4, 5, 6 exons, 1, 2, 3, these, and this is our this is our heterogeneous RNA. By alternate splicing, whether you're in human thyroid cells or whether you're in brain cells, you get different outcomes. In the thyroid cells, they use exon 1, 2, 3, and 4. So they use here. And the translation of that is a calcitonin hormone. It's a small hormone produced in the thyroid that moves around the body to activate other cells in a function. In the brain, you don't really want this hormone here, but what you're trying to do is use the protein itself. And that protein uses exon 1, 2, 3. It skips exon 4 and utilizes exon 5 and 6. After translation, this produces what's known as a calcitonin gene-related peptide. Calcitonin is very important for neural transmission process and how neural cells work. And so it's an important peptide to have there as far as triggering this neural activity. If there's misconnection of these, in other words, you don't get the exact exons correctly made so that the peptide malforms, this can be one form that leads us to migraine headaches. So I don't know if you may have some in your family or not, that certainly know people that have migraines. There are these flashes of light that occur or you can't see parts of things, and then blinding headaches that can occur. They can be brought on by stress, they can be brought on by nutrition, all sorts of things. And there are multiple things that cause migraine headaches, but one of them is an actual missplicing and the production of a malformed protein, which can occur either naturally or by stress-induced systems. And people are learning that stress and certain forms of these stress proteins and things that come from that can actually mess up the spliceosome. So you got a spliceosome that's normal, working fine, and then all of a sudden you got a stressor that comes in and it causes a missplicing to go on. And if you get enough of that missplicing, you can end up with problems. Another thing to tell you about is that there is also this process of what's known now as transsplicing. We'll talk about some of this later, but transsplicing is actually where you produce a message from one gene over here and a message from another gene, completely different gene, and somehow those are combined up to give you yet a full message that's a hybrid of the exons from two completely different genes. And this is known as transsplicing that can occur in this form. So it's and there are other forms of transsplicing, but that's the one that I'm mentioning here. Now, just to kind of give you an idea of what we're talking about here, trypanosine is a protein that's, along with actin, causes muscle fibers and these filaments to function, and it controls the interaction of actin and myosin during contractions, okay? And the primary transcript looks something like this. There are 11 exons that are available. In skeletal muscle, you drop out one of those exons, that's number 2. Skeletal muscle, right? Smooth muscle, like the lining of the intestines and other parts where you have smooth muscle, you actually drop out 2 of the exons. You skip those, 3 and 10. In liver, you lose 2, 3, looks like 7 and 10. And then in brain, where you actually have these fibers that are important for the formation of these neurites and the movement of things along those neurites. You don't use 2, 3, 10, and 11. Okay, so there's clearly, it's the same gene uh, being activated in different parts, different organs of the body, different parts of the body, cell lines, but then the transcripts that are produced there end up being very different, and the proteins, obviously, from those final transcripts being very different. Okay? Now, as I said, it can be a good thing. It can be making, as it was there, can be making these specialized proteins from the same gene that are specialized for each type of tissue that you want. But it can also be problematic if you've got splicing defects, and they can occur either as in this case, primary splicing defects, which means that you simply have a mutation that might occur somewhere here in one of these areas. Okay, so you might have a point mutation at one of these highly conserved areas where you have a signal point that indicates that you're at the end of an exon at the start of an intron, or it could be on the other end, or it could be in one of the lariat forms or whatever. You could end up with a defective protein because you don't recognize the fact that you need to splice out this intron, and you include it. Well, once you include it and it makes a mature message, then we're going to start adding in amino acids that we don't want. You can also, as I said, have mutations, obviously, in the splicing proteins themselves that make up spliceosomes. You can have all sorts of places that it can occur, that you can have mutations. You can have frame shifts. You can have all sorts of things that can occur from this. Just some examples of some of these. There's the lemon protein here is involved in muscle structure, and there are a number of different human conditions that are mutational conditions that have to do with muscle tone and muscle structure and their ability to compress and do what they need to do, ratchet, can be lead to problems, syndromes, and various things. And there's different ones that have been identified by either having mutations that allow these introns to be retained, as is in this form right here, this limb-girdle and allows this. So when it gets read as part of the protein and therefore makes a misfunctioning protein itself. There's other forms, these lipodystrophies, which are lipos is in fat areas that have to do with intron number 8 being retained. There's also all sorts of other forms. I just don't have to worry about too much of them. But the point is, if you get these mutations and you end up retaining an intron, then it becomes an exon in these forms, okay? Or you can actually have a problem where you end up, with not splicing at all, or etc. And you can end up in this sort of having this long-term problem, okay? But there's also, because people start thinking about this, they think about, okay, if we're having splice problems, right, maybe there's ways to therapeutically go after them. And people are testing out some interesting therapeutic ideas. One of them being that you can target for Duchenne muscular dystrophy. There's a problem with some deletion splicing that occurs, ending up with a frame shift problem. And you can attack it with actually by making small pieces of RNA that will go in and interfere with the splicing process and allow you to, that's what this AOS is here. It's basically making you make a piece of RNA and introduce it to the cells, and it allows for exon shifting, prevents this frame shifting, and increases the production of the effective protein that you need. It can occur in all sorts of other places and be used for very fancy things of making these loops and things that are involved with spliceosome formation, etc. And either upregulating, or in the cases here, increasing exon 7 splicing, which is being skipped in this problem. You can actually induce it to be included, or you can form parts where you actually go in and downregulate that splicing so that you get rid of an intron that's accidentally being included, or that sort of thing. Okay? So there are therapeutics, and so those of you that are going into the medical worlds, whether it's in human medicine, pharmacy, or whether you're going into maybe veterinary medicine, these are some of the therapeutics that are being used to attack some of these problems that are known to be genetic problems. All right, so here we are. We've got these transcripts. We've now made this message, and we thought in the past that was the relationship. Well, it turns out that's not the way it works. One gene is capable of not just producing one message, it's capable of producing multiple messages because of splicing. Okay, and I didn't even get to the point where we should talk about the fact that sometimes introns, sometimes they're accidentally used, which is a problem, but sometimes actually they're used on purpose. So introns can actually become exons in an effective way. It's kind of an odd side of it, but instead of just exon skipping, right? You actually use an intron as an exon, and that makes a functional protein, not a misfunctional protein. But what we're talking about here is the fact that one gene doesn't make one protein. It doesn't even make one polypeptide. It makes many different messages, which make many different polypeptides. So really, these people that claim there's a central dogma relationship, it's turning out more and more by the day that there's no central one explanation for what's going on. Yes, genes are transcribed, the messages are matured, they're translated, and we get polypeptides, which ultimately can be combined into proteins. That's the way it works. But how that works, the dogma of that is way naive. There's a much more complicated system that's going on here. And at one level, it's simply this maturing of these messages turns out to be very, very important. And there's a direct relationship in many cases between the splicing and maturing and the transcriptional process, which is guided again by this regulatory control that's up here. We're learning more and more about that every day relative to disease as well as to just normal cellular function. It's a really interesting area for those of you that are thinking about research. Here's an area that you could be really put some ideas and thought into.